Good afternoon, everybody. Welcome you all back to this post-lunch session of today's CME on micronutrients and health. We have eminent speaker with us, Dr. Seshidhar. It gives me an immense pleasure to introduce Dr. Seshidhar KN, who is currently working as professor in the Department of Biochemistry at Sri Devaraja's Medical College, the Constituent Medical College of Sri Devaraja's Academy of Higher Education and Research, Kolar. He completed his MBBS from the same college where he is working, the Sri Devaraja's Medical College, Kolar. He did his post-graduation, that's MD in Biochemistry from Kasturba Medical College. To his credit, he has published 115 publications and he has one patent and also he has published many newspaper articles in state and national level. And he set up a Fluorosis research and referral laboratory in his Department of Biochemistry at Devaraja's Medical College, Kolar. He's a NAC assessor since 2019. He's trained as international lead assessor for laboratory accreditation process approved by APLAC, ILAC to about 164 countries in the year 2019. He has patent in quantitative ELISA method for Serpina 4 calistatin. Welcome you, sir, and over to you to present your talk.
Thank you. Thank you, Kulgari sir, for your nice introduction. I'm audible. So, first of all, I thank the organizing committee, Dr. Satish and his team for giving me an opportunity to say a few things about the fluorosis research, our journey towards the fluorosis research. And I'm extremely thankful to my mentor, Dr. Harendra Kumar sir, who was there with me when we started of thinking of the fluorosis research in our institution. Research in our institution. So the very purpose of we starting the fluorosis research in our institution was that the fluorosis is very rampant in the Kolar district. So it has been observed that the skeletal fluorosis is one of the main component of the fluorosis. So there was a thought process by us, and we had a discussion in the department, and along with my team, we thought of starting and thinking of the non-skeletal fluorosis. The main intention of starting this non-skeletal fluorosis was we started observing the incidence of cancers, incidence of diabetes, incidence of various other non-communicable diseases in the Kolar population. So with this, I went in the year 2017 to Dr. Harendra Kumar sir, who was our dean that time. And when I discussed with him, he told that he extended his full support in establishment of fluorosis research referral lab. Then we thought of extending it, and we did a lot of work on the fluorosis research. Then in the year 2020, our center was conferred with Center of Excellence by the Academy, and sir was the IQS director that time. And we got a funding of extramural funded from the DHR ICMR in the year 2023. And we also have got a research consultancy, and we fitted into all the criteria which are required for the Center of Excellence. So from 2023 onwards, we started catering our services to other departments and also to the other institutions, not only in the Karnataka, even to the neighboring states of Tamil Nadu, Andhra Pradesh, even to Kerala. Then even we had a consultancy from PJ Chandigarh, and also AIMS Bhopal, we had a consultancy with respect to the fluorosis research and other things. So this is my team, so which includes myself, Dr. Muni Lakshmi, Dr. Sai Deepika. These are my main core team who is helping me in getting my work done. And these two are the research scholars, Satish and Sharon Rose, and he is our research assistant who has been appointed for our fluorosis research and referral lab. The main aim of our center was to estimate the fluoride content in biological fluids for management of fluorosis and its complication. And with the objectives that has been displayed here, we started, and with this, let me go ahead. This is the introduction of the fluorosis research and referral lab, how it took birth in our center. The types of fluorosis from morning, many people were speaking about Dr. Bartesh and Dr. So many people were speaking about the fluorosis and other things. So the fluorosis has got impact mainly on the skeletal and dental. So we are working out on the non-skeletal because a lot of work has been done, and there is a lot of controversies and pluses and minuses with respect to the skeletal and dental fluorosis. So we thought of going ahead with the non-skeletal fluorosis, for which we took cancers, we took intelligent quotient in case of children, and also we took so many other diseases, particularly the breast cancer, the cervical cancer we took, oral cancers we studied. Then we also studied on the diabetes mellitus of late in the Kolar district particularly, maybe here also maybe, because this is also fluoride affected area. So the diabetes and other things, when I was a student who was studying, usually diabetes used to be observed. Very rarely we used to observe diabetes. The chance of getting diabetes less than 30 or 40 years was very high. Means the chances of getting diabetes was very less with respect to the people who are less than 40 years. But now it has been observed that even the diabetes we are observing even in 10 to 12 years of age group. And there was a dictum that more than 10 years of diabetes, they used to get microvascular complications. Later on, they reduced it to 5 years of diabetes. Of late, we are observing that these microvascular complications, particularly the retinopathy, nephropathy, diabetic cataract, so neuropathy, all these complications, we are observing it at even after 3 years of incidence of diabetes, chance of getting the diabetes. And it has been observed that the diabetes, once a person is diagnosed to be diabetes, already the disease would have set in maybe 3 to 4 years prior to the diabetes. So this created a lot of interest in us, and we did initially we started with finding out the dental fluorosis and other things in the area. Then it was positively observed that the skeletal and dental fluorosis is very rampant, and as per the Dean's dental fluorosis index, we did a study, and we observed majority of the cases are between the moderate and severe with respect to the Kolar district, particularly in school going age group. This is what we have observed. Then we thought, why not we take up a study to observe these things in multi-organ or multi-system diseases. Then we did it in CNS, so Alzheimer's we studied. Then we studied in Parkinson's disease. Then we studied in intelligent quotient, particularly the government school children and the private school children. We did a comparative study, and even in the adolescent age group also we have studied, and we have observed notable findings, and these things we have published in newspaper articles also. Cardiovascular diseases, we took the atherosclerotic plaque, and in addition to the calcium, magnesium, and various other ions that are available, even we did extraction of the fluoride, and we did a study on the fluoride, and we could observe that the fluoride content was almost on par. The ratios, what we observed, the calcium to fluoride ratio, what we have observed was very narrow with respect to the calcium and fluoride concentration with respect to the atherosclerotic plaque. Renal system, so renal calculus and nephropathy, all those things were very high with respect to the population who were exposed. One deficit that we could not fulfill is doing the case control study, because control group we had to get from a non-fluoride exposed population, which was very difficult for us to get it. So this is one of the limitations of majority of the studies that we have done, either with respect to the CNS, CVS or renal. GA also we have observed, and we gave the recent concept is of giving the alkaline water. So the rat experiments we started with, then we started it with human experiments also. We have observed that the gastrointestinal complications are very high with respect to the population who are exposed to the fluoride water. And then we started giving them with the alkaline water, and with very minimal permissible limits of the fluoride was added to that one. And we could observe that there was a good improvement in cases with gastric ulcers and so many other things. Even the duodenal ulcers, also we have observed a good output with respect to the fluoride research. Hematological, particularly the pregnancy induced hypertensions we have observed, and pregnancy induced anemias. So we have observed. So we could see that there is a lot of linkage between the fluoride exposure as well as the iron deficiency anemia. Even the vitamin B12 and folic acid was also very less. The very purpose that we derived is this fluoride exposure will damage the intestinal flora so that the absorption of vitamin B12 or folic acid is reduced. So this in turn, nutritional deficiencies of vitamin B12 and folic acid we could observe. Even the iron absorption is also reduced despite giving the iron tablets. We could observe that lot of hematological complications with respect to the pregnancy with anemias. Reproductive system, we observed that a lot of infertility cases with respect to the population which are who are highly exposed. Even in the rat experiments, we observed that there is a lot of reproductive related issues. And morning, as our Dr. Sridhar was telling, the coin test. So place a coin on the floor and we should try to pick it up. So if we could touch the floor, it means that we are healthy. If not, so there is a restriction in the spine. So it is because of the sclerosis that is happening with respect to the intervertebral discs and other things. Then chest to chin test and various other tests were also implemented in the field. And the instrumentation that we use for doing all these things is the ion selective electrode, which is supplied by the Orion Thermo Fisher Scientific. There is a video of the same, which we did a workshop on the fluoride estimation. One of the good things with respect to our center is, so majority of the tests that has been done is with respect to the water and the urine sample. So we have done the fluoride estimation in the biological samples such as the plasma as well as serum. And also soil testing we have done as well as cow's milk. We have tested the fluoride concentration. And even in the vegetable, fruits and other things also we have tested the fluoride grown in particularly the local population. 10 kilometers of the radius of our hospital we took, and the vegetables and fruits were procured from the people who are working in our institution. We crushed it and then we dissolved it in water, and then with non-fluoridated water. And then we could see that the fluoride concentration is very high with respect to these things. Then we started expanding it to the horticulture, we expanded it to the bovine, then we have expanded it to the sericulture, because majority of the people, Kolar, as it is known, it is the land of silk, milk and gold. So the gold exposed population, so now there is no gold there. So silica, KGF, it is exposed to the silica and asbestos. So in that population also we have estimated the fluoride. In addition to the silica and asbestos, we could observe a very high concentration of the fluoride, maybe because of the deeper water table that these population are exposed to. And the silk, the mulberry that is grown started yielding very less. So the silk quality and the quantity was both less, which we thought, and we had a discussion with a lot of the concerned authorities, governmental authorities and other things. And we are in the process of doing a mutational studies with respect to the silkworms. And we observed that the fluoride is causing is inducing a mutation in the saliva of the various enzymes which are required for the production of the silk and other things. So that is in the process, and the findings I cannot disclose it right now as it is in the process. So I wish to play a video here. Can you please play that video? The principle, where is it? So this video recording was made when we did one workshop, a national workshop we did, in which around 700 participants were there. Not only from India, even abroad we had. So even from Australia, there were people who attended to this one, from Dubai, and also
selective
from
electrode by Orient Thermo Fisher. Before you could start, we should remove the safety cap given to protect the sensing element. Once remove, open the spout of the filling or open spout of the filling solution and insert the spout into the filling hole of the electrode. This way, fill the solution up to filling hole. Once done, close the spout with one hand, with one thumb, press the cap so as to wet the sensing element and the cone of the electrode. Once the cone and the sensing element is wet, again open the spout, fill the filling solution through the filling hole up to the filling hole. Once done, invert the electrode so as to wet the O-ring. Once wet, invert it upright position and place it in the pH socket given in the arm of the meter. This is the arm, this is the meter. We are going to turn on the meter. We are going to connect the electrode to the point given behind. Once locked, turn on the meter with the button given on the meter. Thermoscientific Orient Star A214 device, which is clubbed with pH. If you once again click the button, the light will be on. Once turned on, wash the electrode with distilled water and wipe it with a clean tissue. Just touch the sensing element. Take care that you don't rub the sensing element. Once rubbed, you will get 0 reading on the meter. Once you get 0 reading, we shall move on to the standardization of the electrode. This green solution is nothing but this 1 ml, 1 ppm fluoride with PSA2 buffer for standardization, which is readily available. When you get the 0 reading, dip the electrode. It should just be dipped inside the solution. Wait for the reading to stabilize. On the meter, the stabilizing should be changed to ready. The value can be plus or minus 0.1 to 0.2 difference. So we should get more than 0.9, between 0.9 to 1.1 ppm. The reading is now ready and the value we are getting is We shall wait till 0.9 and then we can proceed with the next standard. If the reading is still moving on, we can go for calibration. This is an acceptable value, but still I show you the steps of calibration. This cal is nothing but the calibration. Press on the cal button. You will get the P1 with 0, 0. Now we should click on the press on the start button. Once it is start, it will again start reading the sample. Instead, you can press the override button. Once you override, you will be able to edit the value. Then you can give the value that is 1 ppm. 1 ppm, enter. Once enter, you will get the number 1. Once the number is entered, press on the done button. Once done, it will give you the ppm. You should not get back. You should again move on to the next standard, that is 10 ppm standard. Remove the standard, keep it aside. Please remember to wash the electrode after each sample. Again, wipe it with a clean tissue. Just tap on the sensing element. Tap on the sensing element. Now we shall move on to 10 ppm fluoride standard. That is this. Fluoride with PSAP2, that is 10 ppm fluoride with PSAP2. We are going to dip the electrode into 10 ppm PSAP2. It should just be dipped. Yes, once dipped. Again, once dipped, you can tell to accept. You can go for next. Once done next, you will get P2. For P2, you can again give start. It will start analyzing. Can you see it is going more than 9 ppm? It means the instrument is already working good, but still I'll show you how to calibrate this. Again, same procedure. Press on override. Once override is done, it has stopped to 9.3. You press on edit. Once pressed edit, move on to 10 ppm. Yes, enter. You will get number 1. Again, you should go back to 0. Again, enter. Once done, you click on done. Now again, this you need to, first one was 1 ppm and the second one is 10 ppm. Accept both. Once accepted, click on call done. Once the calibration is done, now you should go on to measure. Once it starts measuring, it will again click on the stabilizing will be blinking and the value is shown here. You can wait for the stabilizing to change to ready. Once ready, you can accept. Once it is ready, the whatever value is there that you need to accept, that is 10.3. So we can now tell the electrode is well calibrated and move on with the estimation of biological sample. Now we have done with installation, standardization and calibration of the instrument. Again, after this, we are going to wash the electrode gently. Wipe on all sides. Just gently tap on the sensing element so that you will get 0 reading on the meter. Kindly remember, you should get 0 reading. Once you are done with this, you have cleaned the standardization calibration with the. Let us move on to the measurements. Ion selective electrode by Orient Thermo Fisher. Before you could start, we should remove the.
So these things are already been explained, and the filling solution is nothing but it is the silver chloride or the potassium chloride, which we are using here. So there is a O-ring which we are seeing here. Just below that there is a filling hole. In this we need to fill the silver chloride or the potassium chloride solution into this one. And then we can, first we need to standardize the instrument with the various standards that are available. Usually we take 1 ppm, 10 ppm and 100 ppm. So these are the standards that we are using. So the background voice which you have heard is by Dr. Sai Deepika, who is playing a key role with respect to the fluorosis research and referral lab in our center. So the various buffers that we use includes the TISAB buffer, and the TISAB here stands for total ionic strength adjusting buffer. And the major component of this TISAB includes the CDTA, sodium acetate, acetic acid, and sodium chloride. So these are the ones, and this creates the acidic pH, and this maintains the pH by creating the alkalinity there. And these are the various TISABs that we are using, that is 1, 2, 3, and 4. The number that is given is for the various biological samples that we are using. So for the TISAB 1, 2, and 3, they are specific for the specific biological fluids. So this is the procedure that we are using for these things, and which already has been explained in the video. As I mentioned, the various biological samples that we have measured is in the milk. So Kolar has got a milk union next to our institution. There we went and where the pool of the milk that we are getting. So there we got the milk and we estimated it. And also in the highly fluoride exposed area. So 3 types of water sources we need to take for any fluoride exposed population if you want to do the study. One is from the source, from the borehole, the water that comes out. And the second source is through the pipe that enters into the, now majority of the villages have got RO system in place. So Jal Jeevan, something morning, one of the government official was explaining. So the water that travels through the pipe, so we can see the crusting of the pipe. It is because of the heavy metals that gets accumulated in the pipe. And once it comes out, majority of the metal ions and other things gets blocked there. So the output of the water. So first source from the water that we get from the borehole, second from the pipe that we are getting just before entering into the RO system, and the third source is the water that we are getting it from the RO filtered plant. The fourth source, it is better to estimate and better to discard it. Ultimately, we are leaving it into the soil itself. Again, what will happen? The concentration of the soil fluoride will be double, tripled, and even maybe around 10 times it will be increased. So it has been observed, and the studies have been done in Kolar district, that if we want to filter out 1 liter of water, 60% of that is around 600 ml of the water is removed as the waste. And that water, they say that the commercial people, when they do the marketing and other things, they say that it can be used for washing our floors and other things. So what will happen? Pardon? Yeah, gardening they encourage, but again the concentration of the fluoride will be double, tripled, and 10 times it will be increased. And further, what will happen when we walk with barefoot? The fluoride can also be absorbed through the skin. So a lot of dermatological complications, dermatosis, and other things are more with respect to the people who are exposed to these things. And when this water, the same water, say for cows, buffaloes, and all, for sheep and for poultry, they are not using the RO water. They are using the direct water from the borehole. So this is increasing the concentration of the fluoride in the muzzles of these poultry feed and other things. So this is increasing the fluoride concentration. Again, that is one of the other source of the food that we are consuming. Even the milk that we are consuming also contains the fluoride. So the fluoride concentration has increased more than the permissible limit in the milk. Around 1.5 ppm is the permissible limit of fluoride in the milk. Now it has crossed around 4. So we have given a recommendation to the Milk Union in Kolar to give advice to the farmers who are getting this milk done to use even the RO water for the buffaloes and other things. And practically it is difficult, but once it is related to the health and other things, maybe a policy may come, a government policy may come in future. So this is another thing. The soil and vegetables also we have tested, and it has crossed beyond. The various other sources of fluoride in addition to the water is from the pesticides and the fertilizers and other things they are using. So which contains lot of fluoride. This is giving add-on to the fluoride already that is there in the water. So this is one of the reason why the sources of why the output of the vegetables and other things have decreased in Kolar, number one. Number two, you might have observed the vegetables and fruits nowadays, if you keep it out for a longer time, maybe around 12 hours or something like that, there will be black dots on the vegetables and fruits. This is because of the high concentration of the heavy metal ions that are there. It is not only the fluoride. In addition to fluoride, as we go deeper and deeper, it is the uranium also, the radioactive isotope is also increasing in the water content. So we have also started working out on the uranium. Particularly, there is a Taluka place in Kolar, Mulbagal Taluk. There we have observed the fluoride concentration to be around 10 ppm we have observed. In that area, we have collected the water and we are working out on the uranium also. And we got a positive result. In addition to the fluoride, the uranium was also very high in the soil content of that one, and even the water content of the uranium was also very high. The fruits and vegetables, the fluoride content was very high in these areas. The water content as usual, and the biological samples when we tested, it is the 1 ppm that is permissible even in the blood. So it has been observed that the ppm of fluoride, recently we did one school study in which we have observed around 7.8 in one of the school children. So this is the value so that we have observed. And TISAB 1 and 3 are used for the analysis in the milk. So where the milk pH will be is around 6.5 to 6.7 and the adjusted pH with the TISAB is around 5.3. The sample preparation is almost same that has been explained. And buffer 1:9 we need to use, and TISAB 1 and 3 can be used for milk analysis of the fluoride. And soil and vegetables, we have to use TISAB 4, where the pH of the soil is 4 to 4.8. So such an acidic concentration, it is further worsening with the addition of the pesticides and various other things. So the pH needs to be adjusted to 5.5, and 1:1 sampling needs to be done. And fruits and vegetables that are grown in such a high concentration, we could see a lot of spoilage of these things, and also the size of the fruits and vegetables and the taste of the fruits and vegetables are also changed with respect to the fruits and vegetables that are grown in highly fluoridated water. TISAB 2 and 3, the pH is around 7. The adjusted pH is 5 to 5.6, and so this is for the water. So this is for the water, and majority of the people, they are using this one. And for the biological sample, this is what we are interested with. We are using TISAB 2, where we are using the human serum and urine sample. Even we have started working out on the CSF and even in the brain. So the CSF we have studied, and the fluoride we have observed that it can cross the blood-brain barrier. This is one of the important findings that we have observed. And we got samples from, we have got a referral samples. So for from KMC Manipal. So we did around 300 rat samples we have done. And in those rat samples, in the brain tissue, we have observed that a very high concentration of the fluoride. So for that, we need to use TISAB 2. So this is what we have observed. And the intelligence, so the various rat experiments that we do, that is the rot test and various other cognitive experiments that we do for the rat experiments and other things, are impaired in highly fluoride exposed population. There is a lot of forgetfulness, confusion, and other things are happening. Then we thought of doing why these things are happening. Then we could link it to the vitamin B12 deficiency in fluoride exposed population. As we all know that vitamin B12 deficiency leads to a transient loss of memory as well as a confusion state. So this is what we have observed in highly. And then those people who are given with add-on vitamin B12 and folic acid, so their memory power and other things have started increasing, particularly in the school-going children. So this we did during the COVID thing. So we did a clinical trial with respect to this. So the medicines and other things were sponsored by Jagdala Industries, and we got a funding of around 40 lakhs for that one, for doing that. And in addition to that, we also observed the interleukins and other things, so which improved with giving vitamin B12 and the Mulmina health drink, which you might have seen. So that we have given, and we did a clinical trial. and we got a very good results of that one, and we have published an article also. So the various activities of our FRRL with an objective, we started with research, then we extended it to community health services like school health education and various other things. Then also we have extended our services to agriculturists, dairying, then floriculture, then horticulture, all those things we have extended. And education and awareness also we are creating by publishing a lot of newspaper articles. Now we have included it in the diagnostic parameters of our institution. This is also there in the scope of our investigations in our laboratory. So the test request form of our hospital also includes the fluoride estimation. Routinely, the orthopedicians, the dentists, and various specialties they are working out on these things. And we are also working out on non-healing ulcers. We are extracting the tissue there, and we are observing that high fluoride exposure has got an impact on the vitamin C, which will indirectly inhibit the conversion of protocollagen to collagen. So thereby the wound healing is delayed in this population. So we are including the students who are posted to family adoption program, the first MBBS students who are there with us in the Department of Biochemistry. We are suggesting them to give health awareness and other things, and also collect the urine samples to the families. So each student has been given with 5 families. So their fluoride, their urine samples we are collecting, and we did, and we could observe a lot of increased fluoride levels in these things. So students, first MBBS students are also helping us in carrying out these things. So it is something like both the family adoption program as well as our community health also we are involved with respect to the FRRL. We are conducting webinars on fluorosis, and we made online fluorosis research referral. We have got a YouTube channel in which every month we are adding on some information related to the fluorosis. And we are organizing a collaboration with other departments in conduct of the webinars and other things. So monthly camps, fluoride analysis, campus fluoride analysis, we are doing all the source of water in the campus, in our campus. We are estimating it from the boys hostel, from the girls hostel, and also from the various RO plants that we have installed in our institution. So 5 RO plants that are there installed in our institution, we are doing the fluoride analysis, and we are giving the reports. And also we are conducting a school and a college awareness program. Until now, we have conducted 5 institutions. We have conducted this one. We have published a newspaper article every month, and around 18 newspaper articles, and maybe in another 2 days, we are publishing one more article on GI related thing with respect to this one. And we are conducting workshop on fluorosis every year. So the same recording that I have displayed now. So the team members of FRRL are members of globally recognized fluoride society. So the 3 members whom I have told, we are all recognized by the International Society for Fluorosis Research. So I will show you the certification and other things that is given. And the school and college awareness camps we are conducting, and we are educating them. So it is a mix of government schools as well as the private schools. So the adolescents, the teenagers, all those people we are conducting, and so these are some of the photographs and pics that I have taken. So this is the letter of appreciation that we have received from the various schools and other places. So these are the various webinars that we are conducting related to the fluorosis. And so atherosclerotic plaque and other things, Dr. Yashwant is giving us the sample in estimating the fluoride levels in the atherosclerotic plaque. He is a renowned cardiologist in Kolar. So he is giving us the atherosclerotic plaque after angio. So these are the various newspaper articles that we have published. And recently, on 15th September, I have presented a talk on the medicine apps, which is an online professional forum where researchers and physicians across the globe, particularly the three countries, India, Dubai, and American countries. They will be there in this one. And I have presented on the multifaceted approach to fluorosis, and the feedback was good. And these are the various in-house fluorosis projects that we have completed, around 11, which includes the PG thesis, PG dissertation. So this is the urinary fluoride and anemia and maternal perinatal outcome in pregnancy. This is the one. So from morning we had a discussion on the hematological parameters. So this is the one. And this is the estimation of fluoride and certain one in patients with a diabetic nephropathy. So for this, we have got extramural grants also, and this is with respect to the anemia and pregnancy outcome we have studied. And we have done a field survey in 3 endemic areas of particularly the Bangarpet district we have done. And morning, the prevention aspects people were talking about. So this Moringa oleifera leaves, Ms. Pravalica, and with our team, Pravalica, who is working here in your institution. So we have done, we have written a paper on this one. So Moringa oleifera leaves in the rats and other things. So we have also created knowledge and awareness about the fluorosis in rural population of Kolar district. So on the thyroid hormones, we have done on the endocrine system also on the thyroid hormones. So vitamin D binding protein in diabetic nephropathy also we have done, and this is the PhD thesis work that we have done. So now this work is being carried out. He is a Nepali. So he is conferred with a PhD degree, and he did PhD under me. So this work is extended to Nepal now. So we are planning to establish a center there also in Nepal. And this is the association of fluoride in type 2 diabetes mellitus, microvascular complications. So this we have done with respect to the diabetic nephropathy, retinopathy, as well as cataract. So these are the 3 things that we have taken, which includes the general physician, an ophthalmologist, as well as a community medicine person in this one. And serum biomarkers and fluoride level in the placenta also we have studied the outcome of the placenta of the pregnancy we have studied with respect to, and this is the PhD thesis work. And this is the study, so Bacopa moneri on cerebellar toxicity, arsenic and nicotine on sprayed dolly rats. So on the rat brain, we have studied about the effect of fluoride, and treatment with Bacopa moneri and cerebellar toxicity we have observed, and we could get a good outcome of this one. Histomorphological changes on glutamate levels in fluoride and arsenic-induced injury in hippocampus. On Wister rats, we have done on 60 rats. This study was done by Dr. Suresh, an anatomist in our institution, and recently he has been conferred with PhD degree. And in-house fluorosis projects, role of fluoride and oxidative stress parameters in senile cataract. This is the PhD thesis work of our HOD of ophthalmology. So urine protein to creatinine ratio for evaluation of preeclampsia and its maternal and fetal outcomes. So Ashwini Kashi, she is an MD OBG. Now she is in 2nd year. So we are doing a study on this one. And almost the sample analysis and other things is completed. Influence of fluorosis on genetic predisposition. LOX1 gene we have studied, and this is published. And this is pediatrics related, that is clinical and laboratory profile in pediatric patients with bicytopenia or pancytopenia in a tertiary care center. The work is in progress, and now Dr. Maram Reddy, who is a PG in pediatrics, he is in 2nd year now. He has started collecting the sample, and we are storing the sample at appropriate temperature of -20 degrees. And this is the funding that we have got to a tune of around 20 lakhs. So identification of certain one gene polymorphism associated with fluorosis and type 2 diabetes mellitus. And 1st year we have completed, and we have submitted the bills and other details related to this one. And we are going to start with the 2nd year. And we are planning, in addition to this, we had an negotiation with IIT Chennai. So they are the people who are good in mechanical engineering field in IIT Chennai. So those people, they have assured us that they will get us with a instrument where we can carry it like the glucometer, where the serum sample can be used, and with a drop of blood, we can, with the whole blood, we can do the estimation, and the work is in progress. And we have published around 10 articles exclusively related to the fluorosis, a mysterious mineral, the fluoride, and these are the various articles that we have published exclusively with respect to the fluoride. This is our in-house funded project that we have done. And the consultancy project, we have done more than 300 samples with Kasturba Medical College, Mahe. So Aksa Fatima. And so the title is Role of Betanin as SERTIN1 modulator in fluoride-induced neurodevelopmental. And this is another project that is newly begun. This is another pharmacology with SERTINs in gender-based neurodevelopmental toxicity in fluorosis exposed. This is a preclinical study that we are conducting. With Mysore Medical College, the STSICMR consultancy we did with Mr. Jiva M. Ravi. This is the STSICMR. And Dr. Shubha Jayaram, she is the professor and head of the Department of Biochemistry, MMC, Mysore Medical College. So this is the title that they are doing, and this is the revenue that we have generated from MMC. So the membership, as I mentioned, so this is the International Society for Fluoride Research, where they have conferred the certificates to all the 3 core people who are involved with our FRRL, the Center of Excellence. So they are publishing a quarterly journal on International Society for Fluoride Research, in which this is being published. And the goal ahead with respect to ours is the comparison of diseases with non-fluoride endemic population. We are also doing on SNPs on gene sequencing with respect to the genetic studies, as I have told you. The mulberry and various other things we are working out on that one. Cell culture studies on specific organs also we are working out. So the points of contact for these things includes our YouTube channel, Fluorosis Research and Referral Laboratory. So you can also contact us through the mail that has been published here. So you can reach our campus and have a visit if any of the people wants to or wish to do a collaboration with us, the Fluorosis Research and Referral Laboratory under the aegis of Department of Biochemistry. So once again, I thank my mentor, Dr. Harendra Kumar sir, for supporting me in the beginning stages where I didn't know how to go about it. So when I went and discussed with sir, so he gave certain ideas and plans. Based on that, so I could build up to that one, and it has reached this extent. So thank you, sir, for supporting us in the beginning. And I thank Dr. Satish for giving me an opportunity, and the entire medical college for giving Sridevi Institute of Medical Sciences for giving me an opportunity to speak on the fluorosis research that we are conducting. There is ample of things that are linked with the fluoride. So B diabetes, as we all know that glycolysis, which is the first cycle that we read in the biochemistry, where we have studied that there is a competition between the fluoride and manganese ions with respect to the enolase. Yes, that is true. That is being done nowadays. Lot of research being done, and even we have done a study that a lot of the alcohol, carbohydrate, sorbitol, aldactol, all those things are increasing with the inhibition of this enzyme, where the effect of product is increasing, so that the glucose will be increased and the subsequent products will be reduced. So this glucose will take an alternative pathway and it gets converted into these things. And these molecules, these alcohol, carbohydrates are highly hygroscopic molecules, which are accumulated in various cells of our body, particularly the lens, retina, the Schwann cells, as well as glandular basement membrane, renal cells, all these places it gets accumulated. Since it's a hygroscopic, it absorbs the moisture that is there in the cytosol, it swells, and it causes the effect. So this is the basic pathogenesis that is involved with the fluoride with respect to the enolase. So like this, lot of linkages that has been done, and even it has been observed that the fluoride can bind with the positive ions. As morning, Dr. Sridhar was speaking on the hypertension, which is due to the cations. Yes, that is true. The fluoride, there is a controversy that high exposure to fluoride, they say that it reduces the hypertension. Some of the studies, they say that it increases the hypertension. Increases the hypertension due to the formation of various plaques and other things. Reduction of hypertension because it binds with the sodium and leads to sodium fluoride, and so that the sodium will get excreted. So a lot of research needs to be done with respect to these things. However, we will see how the things will go, and we will take it further. So with this, I thank once again the organizing team for giving me an opportunity. Thank you one and all for your patience.
First of all, I want to congratulate you, sir, for doing such an extensive work in field of fluorosis. And also thank you for sharing your work with all of us. We got really enlightened about so much of work being done in this field. Sir, one question.
Yeah.
That ISC what you showed there, in the same instrument, can we do estimate serum fluoride levels?
Same
also?
instrument, ma'am.
Same.
Only thing is the proton ionic buffer that we are using is the one that changes there.
Oh.
So TISAR changes. Otherwise, the method, principle, everything is same. The same instrument we are using.
Sir, and is there any like advantage of measuring serum fluoride levels over urine fluoride levels? Because measuring urine fluoride levels would be easy, and as they were mentioning in the morning, even in that national program also, that measuring urine fluoride is already included. So if measuring serum fluoride has any extra advantage over this,
Yes,
sir?
Yes. See, thing is, any biological molecule, if you take, they have a renal threshold. Be it glucose, be it urea, be it creatinine, be it fluoride, be it magnesium, any of the ions or anything of that sort. So once the renal threshold crosses, then only it starts appearing in the urine. So by the time it reaches the renal threshold, say for example, glucose, till 180 milligrams per deciliter, the urine, the glucose will not get excreted to them, but it has got its own limitations. The GBM should be good. There should be good filtration. So there should be the loop of Henle and other things should be good with respect to these ions and other things. So if there is any defect in these things, if there is any narrowing, then what will happen? So it gets filtered back and it gets increased in the serum, in the blood. Number one. Number two, by the time it starts appearing in the urine, already the disease would have set in and the damage would have been caused by this. So for that, it is always better to.
For early detection,
Yeah,
it is better to. Any other
Thank you, sir, for a nice presentation and multifaceted research work you have done on fluoride. And also you have extended your research into many other fields like agriculture, sericulture, medicine, diagnostics, and all that. Yeah, really, it's a great and commendable job. I have only one question. Okay, in case of analysis of all these vegetables, food products, how do you select the samples, and are there any specific guidelines for processing of the samples?
We proposed our own guidelines.
Yeah.
Like first we estimated the fluoride level in the water in which it is grown, like the three sources I told you now, sir. So those three sources we have estimated the fluoride. Then in the known population where they are using the same water for agriculture, so that vegetables and other things we took and we estimated. So Kolar, mainly we cultivate in Kolar district. So the potatoes, the tomatoes are the two things which we grow in excess in Kolar. So these are the two things which we took and we estimated. And further we had a doubt whether the lycopene that is there in the tomatoes, does it have any impact on the fluoride levels? So the the flowery area of the tomatoes, there also we have estimated. And this work was done in collaboration with one of the 7th standard student of Kolar, a private school student. So he presented it in Delhi, and we observed that lycopene doesn't have any cross linkages or cross reactions with the fluoride. So this is what we have observed. And once the water concentration has been studied, minus pesticides we have observed, and then after applying, because these potatoes, cabbage and other things, the tomatoes and other things, they're using a lot of pesticides for it. So we usually it is a 90 days crop that is there. So just before they apply the pesticides, we took the flower and we estimated, the fruit we estimated. Then after application of this one, how much of the fluoride is getting absorbed into the fruit, that also we have estimated. And it has been observed that the pesticides and other things which contains the fluoride also adds on to the fluoride that passes through the root to the shoot to the fruit. So this is what we have observed.
Thank you, sir. If there are any questions, you are welcome.
Yeah. Sir, we have not touched that once. Carbides we have not touched because carbides actually it is a banned compound. They should not use it. So when even actually, to be frank with you, when we asked, see, one of the taluka place in Kolar, the Srinivaspur, they are growing a lot of mangoes there, Chintamani and Srinivaspur. When we went and inquired there, they are telling that the vendors, particularly those who buy mangoes and other things, agriculturists are not using. So these are the people who are using it. They told that they are not using any of the chemicals to ripen it. They are using the grass to ripen it. But we have our own doubt. The literature says that there will be uniform ripening when they use the carbide and various other chemicals for ripening the mangoes. But uneven ripening is observed when they do not use anything and when they use the grass. And the shiny thing cannot be observed if they ripen it with the grass. But they are not, as you know, it is the trade secret. They are not disclosing it, sir. And we could not do in depth. We did not go, so we stopped there.
Any other question?
Not sure to for any other purpose, then what can be done for it? Any mechanisms can we do?
Sir, as of now, there is no mechanism for disposal of this biological wastewater. So, like biomedical waste, what we have, there is a set guidelines and other things. So, that itself is a different entity where we can use certain chemicals or certain enzymes and other things, and where we can reduce the certain. There are few studies saying that the RO filtered water can be consumed like the plastics and other things. So there are a lot of research that is being done. So they are trying to find out certain bacteria which can consume the excess fluoride. So maybe those things we may extend it in future, sir. As of now, they are leaving it to the soil.
Because more and more RO plants are coming up every corner with what to do for this? I think seriously.
We have to wait and see, sir, how to go about this biological wastewater.
Can I ask you one more question,
Yes,
sir?
sir, please.
Because we study in biochemistry, this fluoride is an inhibitor of glycolysis.
Inhalation.
In case of fluorosis, there will be definitely higher levels of fluoride. How does it going to impact on this glycolysis?
That is what, sir, we have studied on non-skeletal fluorosis. Because in the Kolar district, we are seeing at a very young age, these non-skeletal fluorosis. Skeletal fluorosis already proved, and people are there with fluorosis. We are with fluorosis. So we are genetically modified of the fluorosis. This non-skeletal fluorosis is the one which created interest, and that is the one which where I went to, sir, to discuss.
Thank you, sir.
That is how we started.
If there are no more questions, we will conclude this session. Thank you, sir. Thank you all.
Thank you.
Thank you, sir, for the session. I now request chairpersons to honor our speaker, Dr. Shashidhar Kain. I now request Dr. M.L. Ayendra Kumar, sir, to hand over token of appreciation to our chairpersons.